In this video, we will discuss about the mechanism of targeted protein degradation. Though we have discussed the mechanisms based on which the targeted protein degradation occurs in the previous paper, but in general, what are the different kind of mechanisms available, or what are the common mechanisms for targeted protein degradations that we will discuss in this video. And they have divided these mechanisms into two different categories. First one is the internal targeted protein degradation, and second one is the extracellular targeted protein degradation. The previous paper we have discussed about the extracellular targeted protein degradations. So here we will not discuss that mechanisms in detail, but we will discuss in general what are the key steps that occurs. We will first start with this internal targeted protein degradation. So in this case, the protein of interest first interact with this small molecule, which is a PROTAC, that is engineered by that is engineered in such a way that one part is interact to the protein of interest and other part will attract the ubiquitination process, which is the internal recycling of the protein process. So in this case, the PROTAC, once it is attached to the protein of interest, it can then attract E2 and E3 ligase, so that they can attach ubiquitin to the targeted proteins or protein of interest, and this process repeats several times, results in a polymerized ubiquitin attached to the protein of interest, and this polymerized ubiquitin then act as a sensor towards this proteasome. And in the proteasome, what will happen, that it will degrade the protein of interest but recycle this ubiquitin, so that ubiquitin can further be used for another protein that needs to be degraded, either targeted protein or the protein that is detected by the intracellular systems for degradations. So this is in general how the internal targeted protein degradation work. And then now we will discuss about this extracellular extracellular targeted protein degradation. In this case, what will happen, the eTPD degrader, or eTPD is the short form of extracellular targeted protein degradations. So this is eTPD degrader is the engineered molecule that can interact with the protein of interest. At the same time, the other arm interact with this degrading receptors. The function of these degrading receptors is that it will cause the endocytosis, that is shown in the intracellular part of this eTPD. And once that endocytosis occurs, then it will release the degrading receptors again to the cell surface, and that is shown here, this recycling or through extracytosis. But the targeted protein or the protein of interest will be directed towards lysosomal degradations. And how this recycling of the degrading receptors occurs to the receptors is still not completely known, but it is known that this thing, but it is the fact that this thing happens, and people have observed this thing many times. And so let's come back to the protein of interest. The protein of interest will be directed towards lysosomal degradation. In the previous video, we have understood that the process by which the degradation occurs is not just lysosome, but it's the autolysosome. And within the autolysosomes, the degradation occurs because of the acidifications. So in the previous video, we have seen how they come to the point that the degradations is because of autolysosomes, as they check by inhibiting the acidifications of the autolysosome process, and they are able to see less degradations of the targeted protein, that is EGFR. So, but in general form, what people see is that the endocytosis will lead to the lysosomal degradations and the recycling of the degrading receptors to the cell surface. And this is the two different mechanisms by which this targeted protein degradation occurs. And in both the cases, what it does is it will utilize the recycling process of the cells. In the first case, they utilize the ubiquitination process, and in the second case, they borrow this endocytosis based lysosome degradations process. So this is how these two mechanisms of targeted protein degradation works, and the design is basically revolve around these two mechanisms. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.